Hello everyone, welcome back to another video of STEMAC, and in this video we're going to talk about translation. So translation is the process in which mRNA gets translated into a protein. So in our transcription video we saw that DNA is transcribed into mRNA, right? But the mRNA that we get after the transcription is a pre-mRNA. So pre-mRNA has some non-coding regions and coding regions. We know that DNA have both coding regions and non-coding regions, right? So when a gene within a DNA is getting transcribed into a mRNA, that mRNA will also have some non-coding region and coding region. So we call those coding regions extron and the non-coding region intron. So this mRNA will need to be processed, right, to move on to the translation level. So that process is called splicing, and the splicing is done by spliceosome. Spliceosome is consists of some proteins and some small nuclear RNA, okay, snRNA. So during splicing, the introns will be removed and the extrons together will give us the mRNA. Now this mRNA, we know that in transcription DNA is transcribed to mRNA and the mRNA is translated into protein. So to get that protein we will need amino acids, right, multiple amino acids. And to do that, the mRNA will first leave the nucleus and they will go to the cytoplasm where they will enter ribosomes, and in ribosome the translation process will take place. So replication and transcription is happening in nucleus and the translation is happening outside of nucleus in the cytoplasm, inside the ribosome. So at first when mRNA will leave the nucleus, they will get to the cytoplasm and enter a ribosome, and this is basically what a mRNA will look like. This is just a shorter form. mRNA will be much longer, but this is just a shorter version of what a mRNA will look like. So here we can see that these mRNA, these are the bases, right? There are 4 bases in mRNA or RNA, which are adenine, uracil, guanine and cytosine. So here we can see that these bases are divided into triplets, right? This is one triplet, this is second, third, fourth, fifth. So these triplets are called codons. They are genetic code and they are called codons. So these triplets are called codons. And each codon will have a complementary anticodon. So these codons, they will each code for a amino acid. So let's say this is AUG, right? This is a codon, and this codon, if we look at this genetic code chart, we can see that AUG is coding for the amino acid methionine, right? So if we look at this chart here, before that we need to know that there are 4 bases, right? Adenine, guanine, uracil and cytosine, and they are forming amino acid. When 3 bases are formed together, those 3 triplets or codons are forming an amino acid. So 4 bases and the triplets, we get 64 codons, right? 64 codon. And from those 64 codons, 61 codon will code for 20 amino acid. Why 61? Because within those 64 codons, 3 codons will not code for any amino acids. these three are stop codons. So these codons, they will signal the translation process to stop it, okay? And we also, if there is stop codons, we will also need a start codon. And AUG is the start codon, but this start codon also codes for an amino acid, which is methionine, but the stop codons do not code for any amino acid. So 20 amino acids from 61 codons. So obviously an amino acid will have multiple codons, right? So if we see here, let's go for this tyrosine, okay? So in the case of tyrosine, we can see that there are 2 codons. Both of these codons are coding for tyrosine. So if we write it down here, UAU and UAC, both are coding for tyrosine. So a single amino acid can be coded by multiple codons, but a codon, one single codon, will not code for multiple amino acid. So this is the concept here of genetic code. So if we look at this here, this AUG will code for methionine, UAU will code for tyrosine, and so on, okay? So now the translation process will begin. So for the translation process to begin, we will need 2 ribosomal units, a small ribosomal unit and a large ribosomal unit. So for the translations to start, we will need the start codon, right, which is the AUG. And at this start codon, the ribosome, small ribosomal unit, will come and bind to the start codon. And now the another RNA molecule, which is called tRNA, will come into the process. So this is a tRNA molecule, and in this tRNA molecule, this is where amino acid will bind. So this tRNA molecule will bring an amino acid complementary to this codon, okay? So their job is to bring amino acids, and at this bottom we will have the anticodons for the codons. So when a ribosomal unit will come and bind to the start codon, a tRNA, initiation tRNA, will also come, and they will come and attach the anticodon with the codon, complementary to that anticodon. So if there is AUG, the anticodon will be UAC, because uracil for adenine, adenine for uracil, and cytosine for guanine, okay? This is our anticodon. And for this codon, we know that it codes for methionine, right? So they will bring methionine, the tRNA will bring the methionine along with it, and they will attach with the start codon. So after this, the ribosome will now move to the next codon, and in this case the similar thing will happen. First we have this anticodon, adenine for uracil, uracil for adenine, and adenine for uracil, okay? And they will bring the amino acid complementary to this codon. So for UAU, this codon, we can see that it codes for tyrosine, right? So it will bring the amino acid tyrosine. Now previously, in the previous step, we already got the amino acid methionine, and now we have got tyrosine. So methionine will now form a peptide bond with tyrosine. Then if we move on to the next step, the first tRNA, the initiated tRNA, will then release from the mRNA. The methionine will stay, and then as new tRNA will come and bind to the third codon, which is AAG, and for AAG the anticodon will be uracil for adenine, and cytosine for guanine. And this AAG codon will code for, let's see, this, right, lysine. So lysine will come and attach to the AAG codon with the help of the tRNA, the new tRNA. So we have now got methionine, tyrosine and lysine, and tyrosine will then form a peptide bond with lysine. Now if we move down to the next step, we can see that this tRNA has also been released, right? And a new tRNA has come and attached with the fourth codon, which is GCA, and for the GCA the anticodon is cytosine for guanine, guanine for cytosine and uracil for adenine, right? And this codon GCA will code for this, right, alanine. So this tRNA will bring the amino acid alanine, and the lysine will then form a peptide bond with alanine. So now we have methionine, tyrosine, lysine and alanine, right? And at the next step, we have reached this codon, which we already talked about at the beginning, that we have 3 stop codons and UAA is one of them. So when we have a stop codon, the process will get a signal. We will get a signal that you have to stop the translation process now. So when they will reach the stop codon, which is either UAA, UAG or UGA, any of these 3 will give the signal to stop the process. So at this codon, the ribosomal units will release, the tRNA will also be released, and at the end we get this amino acid chain, methionine, tyrosine, lysine and alanine. Now these amino acids will go through further processing like protein folding, and after that we will get a protein, which we will talk about more in a later video. But basically this is what a translation is. This is how an mRNA is getting translated into amino acid chain with the help of ribosomal units and tRNA. So that is all for this video of today. Stay tuned for the next video.